Welcome to Spelling Bee Ninja, where learning is not just educational, but also engaging. If you're curious about the fascinating world of science and genetics, you've come to the right place. We invite you to subscribe for our free educational content, connecting a community of learners like you, eager to grow their understanding of the amazing science around us. So, grab your notepad and let's embark on an exciting journey into the life of Gregor Mendel, the pioneer of genetics. Today, we're diving into the remarkable story of Gregor Mendel, often referred to as the father of genetics. Born in 1822 in what is now the Czech Republic, Mendel grew up in a humble farming family. Despite financial difficulties, he excelled in academics, particularly in mathematics and science. When he entered the Augustinian monastery in Brno, his life took a transformative turn. Surrounded by a community of peers who cherished learning, he became a monk and embraced the opportunity to conduct scientific experiments. It's worth noting that Mendel faced setbacks early in his career. He failed the examination to become a certified teacher twice. His examiner remarked that Mendel lacked the necessary insight and precision. But here's where his story becomes inspiring. Rather than giving up, Mendel continued pursuing his research, immersing himself in the vast expanse of his monastery's garden. There, he began meticulously experimenting with pea plants, which would lead to his groundbreaking discoveries. Mendel's perseverance is a wonderful reminder that failure is often just a stepping stone to greater achievements. Now, why did Mendel choose pea plants for his experiments? Well, these little garden inhabitants are perfect subjects for studying inheritance. They grow quickly, produce many offspring, and boast distinguishable traits such as flower color, seed shape, and pod color. Over 8 rigorous years, Mendel studied 7 distinct traits. He examined flower colors, flower positions, seed colors, seed shapes, pod colors, pod shapes, and stem lengths. Through careful control of cross-pollination, he generated pure breeding lines, which enabled him to track how traits were passed from one generation to the next. This extensive dataset, nearing an astounding 30,000 pea plants, revealed mathematical patterns of inheritance that had gone unnoticed until then. Through his meticulous experiments, Mendel formulated the law of segregation, now known as Mendel's first law. This foundational principle states that every organism inherits 2 alleles for each trait, with 1 allele coming from each parent. Now, let's break this down. For example, in a tall pea plant, represented by the alleles T for tall and T for dwarf, half of the gametes will carry the T allele and half will carry the T allele. Through these findings, Mendel's work sparked a remarkable shift in understanding inheritance. Instead of being viewed as a blended mixture of traits, he illustrated that traits are inherited as discrete units. This groundbreaking concept clarified the mysterious nature of heredity, allowing the explanation of why certain traits may appear to skip generations. Following the law of segregation, Mendel also introduced the law of independent assortment, known today as Mendel's second law. This law describes how alleles for different traits are inherited independently of one another, provided the genes responsible for those traits reside on different chromosomes. Picture a pea plant displaying purple flowers and round seeds. The genes controlling these traits can be passed on separately. To discover this law, Mendel conducted dihybrid crosses, involving 2 distinct traits, such as crossing plants with yellow, round peas and those with green, wrinkled peas. He observed a phenotypic ratio of 9:3:3:1 in the second generation, compelling evidence supporting independent assortment. It marked a revolutionary moment in genetics, hinting that inherited traits weren't packaged together but rather existed as individual units. This insight opened the door for even deeper understanding in genetics. Mendel also noticed that among plant traits, some were dominant while others were recessive. A dominant trait is expressed whenever at least 1 dominant allele is present, while a recessive trait only surfaces when both alleles are recessive. Using the pea plant example, the allele for purple flowers, designated as P, is dominant over the allele for white flowers, P. This means that if a plant has PP or PP genotype, purple flowers will bloom, whereas only a plant with the PP genotype will showcase white flowers. The takeaway? The ratio of dominant to recessive traits in the second generation of a monohybrid cross is 3:1, revealing that traits are inherited as distinct units, dispelling the previously accepted idea of blended inheritance. You may have heard of the Punnett square, a grid that aids in predicting the possible genotypes of offspring resulting from a genetic cross. Reginald Punnett, a British geneticist, named this tool after himself. While Mendel himself didn't utilize Punnett squares, his laws lay the groundwork for them. For instance, in a cross between two different heterozygous plants, PP and PP, the Punnett square demonstrates that 25% of the offspring will be PP, 50% PP, and 25% PP, yielding a typical 3:1 phenotypic ratio. It's interesting to note that Mendel's principles have become a staple in genetics education and breeding programs, guiding predictions about various inherited traits, including genetic disorders in humans. Additionally, Mendel's groundbreaking work had an interesting journey. Rediscovered in 1900 by three independent scientists, Hugo de Vries, Carl Correns, and Erich von Schermach, his earlier research laid the foundation for modern genetics. Despite having published his findings in 1866, the obscure journal saw little readership, leaving Mendel's brilliance hidden for years. When his revelations were finally acknowledged, it not only advanced the understanding of inheritance but also changed how traits were perceived in the realm of biology. The interplay between Mendel's discoveries and Darwin's theory of evolution is another fascinating aspect of this story. Both men were contemporaries, yet they never crossed paths. While Darwin offered insight into how species evolved, the mechanisms of inheritance remained unclear until Mendel's work provided the missing link. Imagine if Darwin had explored Mendel's writings, his arguments might have held even greater weight. Today, understanding that genes are situated on chromosomes, we recognize that Mendel's factors are now termed genes, advancing not only our grasp of genetics but also contributing to discoveries in genetic disorders and breeding patterns. Even with the ever-evolving landscape of genetics, Mendel's fundamental principles remain relevant, paving the way for initiatives like the Human Genome Project and modern genetic engineering. Without his trailblazing efforts, the capacity to comprehend, diagnose, and treat genetic ailments such as cystic fibrosis, Huntington's disease, and sickle cell anemia would be greatly diminished. Finally, it's crucial to consider why Mendel's valuable work was overlooked during his lifetime. He published in a niche journal, employed a mathematical approach that left many biologists puzzled, lacked the prestige associated with university affiliation, and challenged the prevailing blended inheritance theory. Ultimately, Mendel ascended to the rank of abbot in 1868, which meant more administrative responsibilities and less focus on research. Sadly, he passed away in 1884, believing his contributions had faded into obscurity. His story serves as a poignant reminder that groundbreaking discoveries sometimes take time to be recognized and appreciated. As we wrap up today's exploration into the life and work of Gregor Mendel, it's clear that his pioneering research set the stage for our understanding of genetics. Through methodical experimentation with pea plants, he uncovered key laws of inheritance that continue to influence how we study biology today. Mendel's perseverance amid challenges, the revolutionary laws of segregation and independent assortment, alongside the significance of dominant and recessive traits, all play a critical role in our understanding of heredity. His discoveries are more than historical milestones, they directly connect to our knowledge of genetic disorders and modern genetic engineering. Thank you for joining us on this enlightening journey through Mendel's legacy. We hope you found this discussion inspirational and educational. Remember, the world of genetics is ever expanding, building on the foundations laid by brilliant minds like Mendel. Stay curious, keep exploring, and don't forget to subscribe for more engaging educational content. We look forward to seeing you next time on Spelling Bee Ninja.